To start off, this chapter focuses a lot on mosquito genetics, specifically about how their genes switch on and off after a blood meal. So there is the concept of the molecular level just after obtaining that blood meal. So this is kind of like a chain reaction that triggers changes in gene expression that impact many things. These things may be things like digestion and egg production. As we have learned in this course, we know that mosquitoes cause many types of diseases. So this image right here demonstrates the many kinds of viruses that are out there as of now. And for instance, this chapter focused on Aedes aegypti as one of the mosquito species they looked at, which is responsible for dengue and Zika. So because of this, it is important to understand the biology behind mosquitoes. And it could also inform us on potential vector control strategies as well. So the first part of the chapter explains how a blood meal acts as a kind of trigger, activating the specific genes in the mosquito. And these changes or triggers aren't random, but more of a targeted response. So you can look at this picture to follow my description, but you do have genes in the midgut, which is where the digestion takes place. Then you can also see the fat body, which is where the egg protein and ovaries are made. So all of these are activated in a precise sequence, so you can think more or less a coordinated effort. So like a system where each organ knows its part. So with this said, you may be wondering how does this happen? How do mosquitoes know when to activate what? So this is where the hormones come into place. So these are known as juvenile hormones, or abbreviated as JH in the chapter, and they play a crucial role. So before feeding, a hormone, that I said juvenile hormone, keeps these things regulated and makes sure the mosquito is ready for the blood meal that it may ingest. And after the meal, the juvenile hormone levels potentially drop, and this signals a whole new phase. And that drop does allow other signals or nutritional cues and different hormones be able to step in and guide this whole process of digestion and development. The chapter then zooms into the protease genes, or trypsin genes that Aedes aegypti and the Anopheles gambiae species have. And the reason for this is that the protease, these enzymes that they break down the proteins, which is important for digestion, and the trypsin genes are abundant and share the expression patterns. So these are good models to study how gene activity is controlled. So you have a more understanding how these trypsin genes are regulated, so you can get insight to the gene expression concept. And it's interesting that the protease expression happens in 2 different phases after a blood meal, so it is a 2-step process. So you can see I have listed that the first process would be your early proteases, that they gear pretty quickly after a blood meal, and their primary role is actually to trigger the production of the late proteases, which are the more or less the ones that do the harder work, I would say. They're responsible for breakdown because they break down the bulk of the blood meal. So they also, therefore, they signal proteases to come. So the way mosquitoes regulate this is by this 2-phase expression, and that could vary between mosquitoes. And when I say the variation is that some will store these early proteases in an inactive form, waiting for that activation. And other mosquitoes may rely on controlling that gene activity to produce them in just the right time. So the 2 main types of trypsin, therefore, are the early, which is produced right after the blood meal, and the late is produced later in a much larger quantities. So you may be wondering, or if not, I'm wondering for you guys, is what happens if you interfere with this process? Well, the chapter does go over an experiment in where researchers added that soybean trypsin inhibitor, and that was added to the protein meals of the mosquitoes. So they had that component that of the molecule that blocks the trypsin activity, and they practically found that mosquitoes cannot fully digest the protein to develop the eggs. And the interesting part of this experiment is if partially digested protein meal with a different type of trypsin before adding the SD1 that they added, the mosquitoes could bypass the block. So this is how they know that the juvenile hormone is involved, because they surgically separated the mosquito midgut from the glands that produced this hormone. So they were cutting off the communication between them. So without this juvenile hormone and isolating of midguts, they could not produce early trypsin. But when then they added the juvenile hormone back in, the early trypsin actually, the production resumed. So they don't necessarily know if adding this hormone directly to the trypsin gene, or if it's from other factors. So this is also why it's important to have more research over this. With the trypsin category, I decided to add this graph from the chapter that goes over the hours after feeding with the early trypsin and the late trypsin. So if we look at the late trypsin, this is responsible for digesting the blood meal, if you recall. So the chapter tightly controlled, but in a different way. And this type and amount of protein in the blood meal seems to play a role, because it's about the specific components within the blood meal acting as that signal. So if you can see that, you can see that the levels of early peak in the beginning and go back down. And then it's kind of the inverse reaction with the late trypsin, as it starts low and then it increases as the hours increase. So you can see that relationship there. That's why that the late trypsin expression peaked after 18 to 24 hours later, and then the early expressed in the beginning. So it's kind of the opposite of each other. With that said, there are other digestive enzymes. For example, the difference between the gene expression between Anopheles and Aedes. I'll start with Aedes aegypti first. They're for the digestive enzymes that are in aegypti are the chymotrypsin, carbonotrypsin, peptidase A, and aminoheptidase. So those enzymes are actually show the gene expression patterns that are very similar to the early and late trypsin, and it has been suggested that they might be regulated by a shared set of genetic switches. So this ensures the breakdown of a blood meal. In contrast, the Anopheles gambiae does have a cluster of about 7 trypsin genes, and they all have different roles. So 2 of those genes, I believe are NTP1 and P2, are specifically activated after a blood meal. And this insinuates that they potentially play a role in digestion. And the other 5 genes out of the 7, they are always active, so the expression does decrease after a blood meal. So the NTP4 from the gambiae is stored in an inactive form, and it gets activated with any food intake. So this could be food intake other than blood. So I thought that was pretty interesting. And 2 of their chymotrypsin genes are induced by the blood feeding, with chymotrypsin like the genes down regulated practically. Another portion of the chapter goes over identifying new genes in mosquitoes and identifying their function. So the entire mosquito genome sequence would help us ask more questions about what genes are involved in things like disease transmission, development, and behavior. So some techniques I want to point out that the chapter went over are things like PCR, the library sequencing, the express sequence tags or the ESTs, and the genomic survey sequences or GSSs. So the PCR essentially would let us amplify specific DNA fragments, like making millions of copies of particular gene sequence. The library screening, I would imagine a library, and each book contains a different gene from the mosquito genome. So it uses a known DNA sequence, such as a probe, to search for similar sequences in the library. So it looks for, let's say, related genes. Then the express sequence tags, or ESTs, that come from actively transcribed genes, so we can see that which genes are switched off. And then ultimately, you have the GSSs, or genomic survey sequences, that are similar to the ESSs, but with random pieces of DNA, giving us the broader view of a genome. So this would all help us with finding genes potentially involved in disease transmission, development, and behavior. One other thing I would like to note is a fun fact that I know we had gone over before with the aspect of dichotomous keys and stuff like that, but I thought it was important, not important, but a fun fact to remember is that mosquitoes and fruit flies do share that common ancestor of genomes that are similar. So in a lot of experiments, they use the Drosophila to guide their research in mosquitoes because they do have that a lot of similarities in common, or just a lot of similarities. So one other thing before I go to analysis types is that the Aedes aegypti has a genome that is 4 times larger than the Nephelis gambiae because of those jumping genes. But with that said, regarding analysis types, the chapter did go over a few kinds of analysis that we have gone over, or reminded me of things that we have done with, or gone over more or less, in chapter 30 with genetics of vector competence. We did cover in that chapter 30 of the genetic marker technology, like PCR, and the internal mapping, so that intervals with the phenotypes. So that reminded me a lot of this chapter with the many types of analysis. So I thought it was pretty interesting to be able to relate the similarities that are still being used, more or less. And so with that said, we do have, the chapter did go over DNA microarray, and that allows us to measure the expression of thousands of genes at the same time. So like massive gene expression, a monitoring system. So I would imagine a slide with thousands of DNA clones, and each representing a different gene. So we can hybridize a fluorescently labeled cDNA from different conditions, let's say, from unfed blood to the slide. So we would be comparing the gene expression profiles, more or less. So this includes analyzing color patterns so we can see which genes are regulated or downregulated. And then we have the chapter going over phylogenetic footprinting, that as a tool for finding the regulatory elements and the transcription factors that bind to them. Then we do have something called DNA I footprinting, and that lets us pinpoint the exact location where transcription factors bind to DNA. The way it works is that it uses the fact that DNA is bound by a protein, is protected from being digested by the enzyme DNase I, and by comparing that digestion, that pattern with and without the protein, we can be identifying the specific sequence where the protein binds. And then lastly on this slide, we have the gel mobility shift assay. So that technique solely confirms the DNA protein interactions. So this is based on the fact that DNA bound to the protein moves slower through the gel than the free DNA. So overall, it is difficult to identify the true elements. So this is why it's important. Research is always important for knowing more about mosquito gene regulation overall. So there are future directions in mosquito genomics. There's the functional validation of genes through the RNAi, And then there's a mosquito gene editing known as CRISPR-9, and there is implications for vector control to reduce these mosquito population, and using this genomic or these genomic technologies to develop targeted insecticides. So the idea of the recent research is the CRISPR-9. I have found some articles that are pretty recent as technology for mosquito eradication, and it has more of the concept if we should engineer mosquitoes to eradicate themselves. So in that case, they could go into areas that they normally live in, and they could interact with each other and basically eradicate themselves. So the way to do that would be the CRISPR-Cas9 technology, and that has already actually revolutionized in many areas of biology and also medicine and agriculture. So where new crops will come from are new cures and scientific discoveries. So you have a protein machine that is guided to a particular gene of the genome by the blue guide RNA that locks into the gene, and then the Cas9 protein cuts it. And so you can destroy the genes and put new genes in. So the idea is that we break the rules of Mendelian inheritance that I know we discussed over, and by using that CRISPR-Cas9 to put a female infertility gene into a population. So if there's an essential gene that female mosquitoes need to be fertile, we would essentially just break it. And we would take a mutant female, and the thing at the top, you mutate this one gene with CRISPR-Cas9. You mutate that with CRISPR-Cas9, and through the degree that gene is copied over into another chromosome. And normally the genes would assort. You would have the mom, the one copy of the mom would go there, but no. And the gene drive mechanism breaks the rules. And so now you end up, every time you have a female carrying that gene, all of their children will inherit it too. So it is an incredibly very effective way to push that sterility mechanisms, not by eradicate them by the billions, but start with a small number of those females that carry this for infertility gene. And after a certain amount of time, well, in theory, by this model, that infertility gene will start to go through the population. So that is what is actually being shown in this slide. So whatever a female inherits, 2 bad copies of the gene, she will be completely infertile. And the male would be fine. It would be able to interact with other females and eventually start to crash the whole population. And in this figure here, you can see that as the line is rising to the right, you have your in your x-axis, you would have your generations, and your y-axis, you see the frequency of added gene. So as the line is rising to the right, those 4 generations of mosquito is about, I would say, 2 months. So in, let's say, your 2 months, it appears you should start to see this whole population replacement, where all of those mosquitoes that were born, the females that are sterile and the males are carrying a female sterilizing gene. So it has that probability that it's working in the lab, but you would have a lot of benefits. And we would be able to send mosquitoes to murder themselves and eradicate their own population. So we did see the benefits of what CRISPR-9 could do. However, you might wonder what could go wrong. So it sounds great, so why not do it now, right? So some of the things that you can consider is that we don't know what's going to go wrong. So when you delete a species, it wouldn't just be one, it would be about 30 species, because this isn't just one disease vector mosquito that is present. So you eliminate a lot of that insect biomass that animals like bats and birds, for example, eat. And they could find other sources, of course, of food, but it would still be a major problem because I don't know if you've ever gone out around dawn. You could, and well, at least in Austin, there is a bridge, and you can see that bats do clear a lot of mosquitoes from the air, and they will go hungry if it's taken out of the system. And not only that, but secondly, you can see that mosquitoes are pollinators. They're known as pollinators to an extent. But so you can see this is a male mosquito, which they don't bite us, but the male mosquitoes drink this nectar from flowers, and they have limited roles as pollinators. Although we may not know, but what if they serve a major role as pollinators of plants that we didn't anticipate before until we eradicate them? Then we would have a problem that can be reversed. And then another issue that we could think about is we have no idea what happens if you take that CRISPR-Cas9 gene drive system and drive a female infertility through a mosquito population, because we're not for certain that will jump to other insects as well and affect them. Last but not least, this chapter did go over a lot. Something I wanted to add is that there are other ideas out there to take a death gene, for example, that is very effective and kills mosquitoes efficiently if you don't want to mess with the infertility aspect that I explained with CRISPR-Cas9. And you would just kill them, and you can use that gene drive, that's what it's called, to drive a death gene called HID, I think, or HID. So the HID is I'll call it HID. HID is basically a death gene that kills the cells. So it would kill the cells very efficiently, but there also isn't a guarantee that gene drive mechanism that's loaded with a lot of HID will not go out of hand. So the thing is that with research at least, is that we are at the point of incredible technology, and it functions perfectly in lab, at least from research articles. But on one hand, you have animals that are killing, like we discussed in class, so we're killing hundreds of thousands of people every year, and they sicken hundreds of millions in many sections of life on this earth. And we have the promise of technology on the other hand, just to delete these animals or vectors. But with everything and anything, there are risks. So it just is a matter of decision of how it would affect everything overall. But other than that, this chapter was a really nice read. I think I read it like 2 times because I just wanted to fully understand it, and I thought it was cool reading it. Not biased. But this chapter did go over the mosquito gene expression and how it is a very important process, the blood digestion and reproduction aspect of it, the hormonal regulation of juvenile hormone. That's actually found in many insects, not just mosquitoes, by the way. And then the genomic techniques that are commonly used, and how future research is very important and can help us control many mosquito control strategies like we have gone over throughout class. So that is all I have for today. I look forward to discussing with you in class. That said, thank you.